I'm Wayne Poon from NeuCyte, where I am the head of neuroscience. And today I want to share with the audience some work that we've been doing using KIF1A patient iPSC-derived neuronal models, in which we will be I will be describing our recent drug discovery efforts. Okay, so what are iPSC models? They are induced pluripotent stem cells. They are derived where we collect fibroblasts or blood cells from a patient using the Yamanaka factors, 4 factors. We can reprogram your cells to human-induced pluripotent stem cells, which then have the potential to differentiate into any cell type in the body. Over the years, there's been quite a bit of advances in defining the methods on how to generate the different brain cells. And at NeuCyte, we have the ability to generate many of these. And what we do with these brain cells is we build 2D brain in a dish models to study mechanisms of neurodegeneration like Alzheimer's disease. We can incorporate these different brain cells into 3D organoids or assembloids, and to also better model the brain in a dish. And using these platforms, we can collect some phenotypic data that will allow us to screen or uncover pathways for drug discovery. So the SynFire platform, which incorporates the generation of trans-differentiated neurons, and here we just are showing some of the many assays that we have developed to phenotype these cells, including imaging, patch clamp, electrophysiological recordings, and then also using imaging to assess phenotypes, over time, such as a NeuCyte and synaptic development. So one of the platforms that NeuCyte has developed is using electrophysiology, and specifically using microelectroarrays, where we plate the brain cells on a dish here that have 16 electrodes per well, and then over time the brain cells, which include the neurons, will develop and then spontaneously fire. And then at NeuCyte, we have both medium throughput, and then we also have an automated system where we can really scale up this MEA phenotype for drug discovery. So the SynFire technology, which is where we're able to generate induced excitatory and inhibitory neurons for drug discovery, is because when we make these neurons, we can make these neurons with essentially low lot-to-lot variance. What we observe is consistent network maturation from week 1, 2 to 3 here, and then looking at phenotypes such as weighted mean finding rate, you can see the consistency of our neurons plated in the wells of the MEA plate. So with this SynFire platform in hand, we've been able to model things like what's shown here in this panel is epilepsy or inducing seizures in a dish, and we'd be able to test 18 different compounds that show good correlation in terms of the concentration needed to prevent seizures in our model and the clinical data that others have obtained. So with this in mind, NeuCyte and KIF1A.org entered into a partnership here where we applied the SynFire technology for CAND, in which was facilitated by KIF1A.org, in which they had patients with KIF1A mutations. They were reprogrammed to these induced pluripotent stem cells, and through this partnership, we were able to obtain these stem cells, and we generated neurons from these patient lines. And shown here is both phase contrast and immunocytochemistry of neurons from our wild type control that we have at NeuCyte, and then additionally the patient iPSC neurons from an E253K patient and a P305L individual. So with these neurons, both excitatory and inhibitory, we're able to apply these cells to various phenotypic assays to uncover phenotypes that we can drug screen. And so some of the things we looked at are axonal outgrowth, NeuCyte phenotypes that incorporated some microfluidic devices. We are beginning to do some of the time-lapse analysis to see how spines develop over time. RNA-seq to identify novel pathways that aren't readily apparent from KIF1A mutation motor deficits. And then we are in the process of developing a proximity ligation assay to examine KIF1A mutant interactions with microtubules and how they might be modulated by tau. One phenotype that we uncovered, and I'll go into this in more detail, is that KIF1A mutations exhibit a hyperexcitability phenotype, and that this phenotype can be taken forward for drug screening efforts. So in partnership with a microfluidics company out of Canada called Inuvio, they have these microfluidic devices that allow us to isolate different compartments. Here we culture the neurons in one compartment, and they're able to grow their axon or their processes into the second compartment. We can adjust the volumes within each compartment to essentially restrict molecules to one side, in this case, either the neurotrophic factors BDNF, NGF, or NT3. And because of this volumetric difference, the molecules in this compartment cannot flow into the axon compartment. So the only way that these molecules get into the cell bodies of the neurons is by active transport being taken up by the axons projected by the neurons. And in this way, we can interrogate mechanisms of trafficking and how the KIF1A mutations might affect this. And so this is just a representative example of the device in which these are then axons that grow through the device, and they are imaged with an antibody to beta-3 tubulin using some of the software that can analyze the neurites or the axonal outgrowth from these devices. We were able to quantify and identify a specific phenotype in which the KIF1A mutations exhibit impairment in BDNF-mediated axonal outgrowth, shown here. Secondarily, we have the tools in-house to carry out some NeuCyte analysis to identify additional KIF1 mutant phenotypes. In this case, this is a sample image of neurons. There's a way software that allows us to classify and identify nuclei, subsequent map to NeuCytes, and then the NeuCytes that express KIF1A. And in this slide, we have identified a specific KIF1A mutation phenotype within P305L, and that is examining the NeuCyte intensity of KIF1A in NeuCytes relative to soma, which is the cell body. And this confirms data obtained by other groups in which the KIF1A mutations exhibit impairment in how the motor protein actually traffics into processes. And so the ratio of the amount of KIF1A in the NeuCytes relative to the cell body seems to be a robust phenotype. We have a Z-score of 0.65, and that is a good score for an assay to move forward for drug screening. Additionally, we have looked at calcium imaging, and in this example, we have compared the wild type to the E253K, and if you can see the synchronous calcium traces here in the wild type, but in the E253K mutation, you see quite a bit of asynchronous calcium firing. And when you sum up the traces, you can see that the wild type relative to the mutant has asynchronous and lower average amplitude of calcium due to the asynchronous nature. And this is, again, another phenotype that we can move forward for drug screening. So getting back to some of our synchronous MEA phenotypes, what our electrophysiologist Peng Zhao has done is he noticed that upon a subthreshold treatment of picrotoxin, which is to induce a seizure-like activity, you can see that with the KIF1 mutations exhibit an increased hyperexcitability shown here after 15 minutes. And so this is just the normalized network burst frequency shown here. And then again, the E253K mutation shows a more exaggerated hyperexcitability. Now, the other thing that we looked at was another metric of electrophysiology, and in this case, looking at the number of the time before electrodes become active again, and you can see that in the E253K and P305L mutations, the networks spontaneously fire more readily than control. And again, these are Z-factor scores that can be utilized for drug screening moving forward. In the future, we want to interrogate the mechanism of some of this hyperexcitability. And there are some studies suggesting that the axonal initial segment, which is a region of the axon at the very beginning that protrudes from the cell body plays a role, or KIF1A plays a role in establishing the AIS. And so we think that the mutations may impair the AIS function, and that this is what drives the hyperexcitability. We are pretty excited that we are going to be developing assays to potentially look at this, and that could be then utilized to uncover drugs that might reverse this phenotype.